OK，这个各位老师，各位同学，大家晚上好。这个肖老师那边上午好。这个我们跟往常一样，今天这个也是非常高兴邀请到马里兰大学的肖春元教授来给大家做报告。这个我来看一下。肖老师现在在这个马里兰大学的这个 College Park 的生物技术研究所做和那个植物科学和这个景观建筑系做教授在什么时候？18年还是19年？18年好像应该是18年对吧？18年我曾经去他们那群访问，肖老师还有他的好几个同事有个非常愉快的交流这个对于他那个系其实有很详细的了解。肖老师他是在中国在华中农业大学获得了园艺的硕士学士就是学士就本科从华中农业大学毕业1984年，然后87年读了硕士，然后92年读完博士，然后中间应该在华中农大工作对吧？
对。
然后到3年去美国佛罗里达大学和这个英国的这个 East Anglia 大学做博士后研究。那么2003年加入University Maryland 以后呢一直在马里兰大学工作，从事这个植物的抗病和抗病机理的研究我刚才还在这个跟肖老师讨论这个肖老师从2003年02年左右01年的一段时间从这个拟南芥里面克隆的这个 RPW 基因，我这样一直问他是不是第一个抗病基因还是抗白粉病的基因没记错了应该是抗白粉病的基因这个很早的时候我就读过肖老师的文章发在 Science 上面，我说哇不得了，这个文章做的特别漂亮，而且是非常早，在我记得是2001年对吧？
对的，
对我这个还是很好。然后这个很多年之后我回南京农业大学兼职的时候，2009年2010年那一段时间我一直就把这个肖老师的这个文章从我脑子里面存了很多年然后翻出来再重新开始做这个有关这个葡萄和这个草莓里面的这个抗白粉病的这个性状包括和它相关的这个MLO 这个就说是一个另外一个是一个 Mutant， 它是这个基因它会导致植物的就是感病，所以这个里面呢就是有很多非常复杂的这个问题。我们曾经做过 RPW 这个基因的这个起源，它的这个基因机构本身是从哪儿起源的我们也在 Scientific Report 上面发过一篇文章，可惜那个时候 Scientific Report 发表的时候还挺有影响，过了2年在中国这个名声扫地，所以我们这引用还挺高，那引用我都没看，但至少有好几十个了，就40-50个可能但是呢就说这个期刊本身这个名声扫地，所以今天非常非常高兴来听肖老师讲这个 RPW 基因和 MLO 这个基因之间的这个关系，他是当时如何来做这个东西。我和肖老师交流过，是肖老师还很想和大家交流一些在这个做这个基因的过程当中的一些个思路和思维方法，和如何去做科研，如何去写好论文所以这个是大家今天是说是这个一箭双雕，但是今天大家来一听二收这个听肖老师讲如何去做这个研究，如何去写好 paper， 然后如何去思考 这方面的问题。所以下面请夏老师来给大家做报告。
OK，谢谢陈老师的介绍，这个够讲够预了啊。让我羡慕我的 screen，OK，Oops。So, Chen Laosi, thank you for your invitation. So I've been thinking about which language I should deliver my talk today. And after some discussion with Professor Chen, I think I prefer to use English because I think maybe some of the people in the audience may not understand Chinese. But I believe everyone here should understand English. So I think probably it's fair, as actually it's probably better to for me to deliver the talk in English. And because the way I speak, I think it's still a typical Chinese style. So I'm sure that most, if not all people, can easily understand what I'm talking about, okay? So thank you very much for the introduction, Professor Chen. I would like to give another kind of different version of my self-introduction, which is the one that I gave to my students in the classroom in the USA. Excuse me. So my family name Xiao, literally, I think means, or at least is connected to autumn. 萧瑟秋风，无边落木萧萧萧 means autumn. Autumn, of course, we know it's beautiful. I mean, really, the beauty there. My middle name, Shun. It's quite difficult for American, for foreigners to pronounce. But it means smooth, right? And also my last character, Yuan, we know it means really the very beginning, the origin. And of course, its pronunciation is similar to another Yuan, means a circle. So as you can see, my parents, they wished that my life journey it was smooth. And to some extent that it was true, but not always. Overall, I think my life is quite smooth, but it does not mean that I did not have frustrations, setback. But overall, I went through all this up and downs, and I have been able to achieve what I have today. I'm a professor at the Institute for Bioscience and Biotechnology Research and the Department of Plant Science and Landscape Architecture at the University of Maryland. But 40 years ago, or more than 40 years ago, I was a cowboy. It's literally a real cowboy in a poor, remote, mountainous countryside in Hunan, Shaoyang province in China. Using Google Earth, you can really identify, where you are with a very high resolution. Here we go. I can even see the map. My house is located there. And here's the river that I used to swim when I was a kid. So it's amazing resolution. But when I was a child, really, I did all kind of hard farm work. I even had hard time to really feed myself. So we, family, or just like all other people in the village, basically the whole country, right? suffered food insecurity to some extent. And also, I remember I really suffered chronic malnutrition. And then was why when I was 16 years old, by the time when I actually took the national exam and passed it, I was the only one who, in the whole entire high school, got the chance to go to college, at least qualified to go to college. But I weigh only 65斤, it's about 70 pounds. And apparently, due to that reason, I was also very short, no college there to take me. So that was a setback, but I continued my high school education. And the next year, I grew a little bit more and got into Huazhong Agricultural University. And I remember when I was about 11 years old, I had to collect firewood from high in the mountain. We live in the at the base, at the foot of the Yunshan, the mountain. I had to collect the firewood with my brother higher up in the mountain. And one time I remember we spent more than 10 hours in the mountain, and we were so hungry. We did not have any strength to come down. And luckily, we found some wild fruits. And you can see here, now maybe more than 25 years later, a young professor, Dr. Zheng Yunliu from Huazhong Agricultural University, visited that mountain and he found this wild fruit. And as you can see, you know that. We call it tengli in our local language, tengli. Pretty sure you know that it's kiwi. It's wild kiwi. So we know kiwi originated in China, probably including Hunan and Wugang province. And I have to thank the kiwi fruits to really sustain me and, give me the energy to keep working. And now, time flies. I graduated from Huazhong Agricultural University. I got all my degrees. And then I did postdoc at the University of Florida in the USA. And 1 year later, I offered a position postdoc in the university. So I had a chance to switch from citrus genetics and breeding to understanding molecular mechanism of plant resistances. So I did not hesitate. I took the position. I went there 9 years. I came back and took the faculty position at the University of Biotechnology Institute. This is our actually institute building, the small campus, but it's beautiful. Well, there we go. We need to rename it. We organized this institute, IBBR. My laboratory here aims to understand and exploit most of plant disease resistance using the Arabidopsis and powdery mildew interaction as a model pathosystem. So when as a scientist, I plotted my career development against time, and I found lots of, it's a curve, of course, but overall the curve is, it's going up, which is good. But now probably I reached the top of the hill. I'm going down for sure. But when I look back at my career, I found lots of key points, or you can say it as a stepstones. The most critical one is the time when I graduated from high school. I got a chance to go to college. So imagine if I had not had that chance, I think I would have been still in the countryside, just like my family, my brother, my sisters, they are still there, right? So that was not easy. And I had a simple dream by that time. As, I wanted to be a middle school teacher. And of course, my dreams kept changing constantly. And the time I dared to dream to go abroad, and dared to dream to establish my own laboratory. And more or less, all these dreams came true. So I'm old enough maybe to give you guys some 心灵鸡汤. So I think if you have these 5 elements, anybody can be successful. First, you have to have a passion. You have a small dream. That dream can maybe becoming bigger, and keep that dream. Okay, so you have passion. No matter what you do, if you have passion, you can do it better, or even better than others. And as a scientist, I think you really should have curiosity. You want to find the bottom of a phenomenon, right? And of course, you have to be persistent. You want to get it done and stay with your, aim. Be focused on that. And of course, you have to be working hard, because nowadays there are so many college graduate students and professors. They are far smarter than you, right? Or you are very smart, but it is still not enough. You have to be working very hard to compete, to win the competitions, right? And of course, you also need some, good luck. Otherwise, your hard work may be screwed up by others. You've been working on a project for 10 years, and just before you want to publish it, and you really found a paper published in Science and Nature, then you get it screwed up. So you need some good luck as well, right? So if you have these 5 elements, your young generations, I think, have a much better and a higher starting point than I. So that means everybody, if you do these 5, you can reach higher, much better position than I do. So I truly believe you can get there, right? All right, now I want to share my story with you today. As I briefly mentioned, that I use Arabidopsis and powdery mildew as a model system. Why? Because powdery mildew is probably one of the most widespread and important plant pathogens. And this powdery mildew fungi belong to ascormicate fungi in the order of Arabidopsis. And they have more than 10,000 species as hosts, okay, including staple crops like wheat, barley, and many of our horticultural or ornamental plants like tomato, cucumber, rose, apple, strawberry, grapes, you name it, okay? So now this is Arabidopsis plants infected with powdery mildew at 15 days post inoculation. You can see it's a thick layer of white powder. So now you can probably understand why it's called mildew, powdery mildew, because basically it's powder, right? Just like snow, like powder mildew. Pretty beautiful, isn't it? So, for some reason, I got connected with powdery mildew, and this organism gave me the passion and the science, and give me the job, give me salary. So I really have to thank Powdery Mildew. But my life dream is also about Powdery Mildew, but it is to defeat Powdery Mildew. I want to kill Powdery Mildew, wipe out Powdery Mildew as an organism. Of course, that's maybe impossible, but that's my dream. I've been trying to figure out a way to help plants to defeat Powdery Mildew. Now, if you want to defeat an enemy, you want to understand the enemy, right? And of course, in this case, the host is not human, but it's plants. So we have to help plants to defeat the enemy. Now, so let's look at the infection biology of a Powdery Mildew fungus. When spores land on leaf surface, the spores will germinate within 6 to 12 hours. And then in 2 days, it can develop the germination tube and mycelia, and then it would differentiate Aprisorium, which will penetrate the plant cell wall and develop a feeding organ called the hostorium. Now, the hostorium looks like just a strawberry, but it's inside the plant well. Everything else of Powdery Mildew is in the leaf surface, not within the plant well. And of course, as a feeding organ, the hostorium is critical for the success of the Powdery Mildew. But being the only fungal structure in the plant well, it is the only target for plant defense as well. So, to some extent, maybe it's not a very accurate analogy. The hostorium is the Achilles heel, the Achilles 之重. All right, so within a week, the successful Powdery Mildew sporulin were developed into a big colony, producing tens of thousands new spores that can, of course, then be spread around and cause new infection. So when I did 1-year postdoc in Florida, I got a chance to go to England, and my supervisor, Dr. Fred Gamita there, tried everything to persuade me to stay with the citrus project. I told Fred, I only had one condition. That is, if you can convert citrus into Arabidopsis, then I stayed. But of course, that is impossible. He's alarmed. Forget about it. You go. But remember, anytime if you want to come back, you're welcome. My lab door is always open to you. So I took the project in England and spent 3 months and to map a gene that confers resistance in Arabidopsis in this accession called MS0 to Powdery Mildew. I found out the gene mapped by a graduate student was not correct. And so there was not such a gene in that, sort of a map location. So I told my supervisor, Dr. John Turner, I said, John, if you believe me, then please retract your paper already accepted by the Plant Journal. Then I continue this project. Or if you don't believe me, I'm going to quit my job. I go back to University of Florida. John Turner believed me. He retracted the paper. I continued the work. I spent 3 to get the gene initially mapped to chromosome 3, but the original gene was mapped in chromosome 4. So John joked with me. He said, "Hey, Xiao," everybody there called me Xiao. Well, it's not a big deal. It's very close, right? It just jumped from 4 to 3, so very close. So, of course, if you are a geneticist, you know what it means. So I spent many years to get the gene clone. It was very, very difficult because the pathosystem, but then the pattern was very weak. I had to use microscopy to do all the phenotyping thousands of F3 families. And also there was a group at Stanford University with a better, more aggressive powdery mildew. So, on such a scenario, I got a gene clone first, and I found out that gene locus consists of 2 genes in MS0, but these 2 genes are missing in the susceptible accession, the model, really the sequencing, the model with this genome sequence, column 0. And column 0 contains 4 homologs of RPW8 genes. Okay. So by that time, I spent years to try to understand how RPW8.1.2, or just called RPW8, confers resistance against powdery mildew. But in the meantime, during the last 2 decades, the entire community worked very hard to elucidate the molecular basis of plant innate immunity. Now, this single slide shows, how plant immune system works. It consists of 2 branches. So when a pathogen, such as a powdery mildew spore, germinates at the leaf surface, it tries to penetrate the cell wall, but something conserved from the pathogen, we call it the pathogen-associated molecular patterns, will be recognized by immune receptors at the surface. So in this case, chitin, which is a unique chemical from the fungal cell wall, that does not exist in the plant well. So plants can recognize as a non-self, and then it will activate the downstream signaling cascade, including the MAP kinase signaling, leading to the defense gene expression. And then you have the so-called Parametric immunity, PTI, right? And then small, bacterial pathogens or fungi, they figured out a way to further invade. In this case, powdery mildew differentiates, hostorum. Now, the hostorum can secrete fungal proteins, effect proteins, into the host cell, and these effect proteins can, of course, suppress PTI and then cause disease, thereby establishing the host pathogen, relationship forever, unless one of the interacting partner dies. And of course, plants, evolved another sort of branch of immunity called we call it effect-triggered immunity, which is basically due to the activation immunity by the intracellular immune receptor of this, we call it NP nucleotide binding site using rich repeat family proteins. These proteins can detect the presence or activity of the effectors and trigger defense response. So some of the go to the nucleus to activate defense. Some actually will be mobilized to the plasma membrane to trigger maybe an calcium influx and causing program cell death. And the outcome, of course, is effect-triggered immunity, which is accompanied with the hypersensitive response, which is program cell death of this infected cell, which of course will generate some mobile signal to activate systemic acquired resistance. Okay, so now this is sort of framework of plant innate immunity, and RPW8, is an atypical resistant gene. So we have been wondering how RPW works, where RPW works. Okay, and during the past 2 decades, our group actually figured out the RPW signaling pathway. We found out EDS1, PAD4, or EDS5, or CYP2, or salicylic acid, or including NPR1. They are all required for RPW8 mediated resistance. So looks like this signaling pathway is the same as the NNRs containing the tier domain. So it's kind of interesting, right? And also we found some other components, 1433, which serves as a positive regulator, or PP2C, phosphatase, or EDR1, MAP kinase, and ERH1, which includes inositol-phosphorus ceramide synthase. They play a negative regulation and regulatory role to control sort of RPW defense in the naive plant cells. So now, as I mentioned, that the NNRs belongs to a superfamily of plant immunity proteins we call R proteins, and they can be subdivided into the one that contains CC domain, CNL, or containing the TI domain, called TNL. They function to sense, to recognize a specific effect proteins we call avian factors from pathogen. So they confer specific resistance to a particular strain or a few strains of the pathogen containing that recognized effector. Now, the discovery of RPW8 now contribute to the finding of another subfamily. Now with this we call RPW8 domain containing NNRs. Basically, from the first day when we cloned gene, I did blast. I actually a long time ago, more than 20 years ago. So now, this is going to be a long story, but keep it short. We found out, or the community found out, that these RNL proteins, they actually are very ancient. Actually, Professor Chen, he briefly mentioned his work actually contribute to the origin and the evolution of the RPW8 genes or RPW8 domain containing NR genes. So it turns out that the RNLs serve as the downstream conserved signaling components for the classic NRs that function as a sensor R proteins. Okay. So in recent years, this area containing this RPW domain NNRs is very hot. Jeff Dangle also called the name as hyper NNRs. Basically, they in Arabidopsis belong to this unique subfamily containing NRG1-like and ADR1-like. Okay, and together with these people, Detlev Wigger in Germany, Jonathan Jones in England, and Xin Li in Canada, and also Brian Stask at UC Berkeley. They now elucidated the function of these RNLs. They found out, as I just said, they function as signaling components. This is a cartoon made by Jonathan Jones group showing that for the ADR1 and NRG1 function downstream of a large array of the NBSLR sensor proteins that can recognize specific valence factors. Okay, and so this RPW8 containing NRs really function as a hub downstream of the classical R proteins. To them, they are essential for immunity in many, if not all, the contexts. Okay, so now recently, Jonathan John, collaborated with me, trying to address the question whether the RPW8 genes, like homolog RPW8 in connectome 0, whether they are required for function. And so his group, deleted the whole RPW locus in Columbia 0, and our group tested the RPW8 now mutant with first with adapted powdery mildew. And you can see the Columbia 0 is susceptible, but the RPW8 now mutant is more susceptible, as shown by quantification as well. So that means RPW8 homologs, they contribute to basal resistance, right? And we also tested these mutants with, an non-adapted or poorly adapted powdery mildew. We found that the now mutant can support better growth of that non-adapted powdery mildew. So further suggesting that RPW8 homologs contribute to basal resistance. And also Jonathan Jones' group showed that the RPW now mutant actually is more susceptible to virulent bacterial pathogens as well. But the now mutant has just normal ETI resistance against the avirulent bacterial strain. So suggesting RPW8 homologs do not contribute to effective immunity, but somehow it contributes to basal resistance. And then recently, Shunyuan's group at University of British Columbia knocked out all the 6 genes, okay? And she called it a herpes mutant. And the herpes mutant is super susceptible to bacterial pathogens. And then our group got this double mutant and the triple mutant as well. We tested it with powdery mildew. We found out that ADL1 triple mutant, all the 3 ADL1-like genes are knocked out. Now, the triple mutant is super susceptible to powdery mildew, to the well-adapted powdery mildew. The NRG1 triple has a little bit of enhanced resistance, susceptibility. But the six-double mutant, okay, with all the 6 RPW8 containing NRs knocked out, shows the highest acts like a molecular mixer. It senses where the interface is and then activates defense right at the interface. So now, being in this mysterious or uncharted sort of territory, the host pathogen interface, we got a lot of interesting questions to ask. But due to time constraints, I'm going to just talk about a few here. So from the very beginning, we wanted why RPW goes to the interface. So how first? So women did a large-scale mutational analysis on the RPW gene with a lots of more than 100 deletion, mutants, and figured out these two short domains are absolutely required for RPW targeting to the interface. And also he did a so-called Nairs scan, replacing a small stretch of, a few amino acids in the RPW gene, and then screens which region is more important. Again, he found the same domain, same motif. These two are absolutely required. Interestingly, these two motifs contain basic residues with a pattern, like either lysine or arginine, or another lysine or arginine. So we thought this RK
recording stopped.
appears to be post-code or zip code to the interface, and indeed made a single substitution, amino acid substitution in the first domain and the second domain for these two motifs. These two substitutions abolish the RPW targeting to the EHM, the interface. So suggesting these two motifs are absolutely required. So now the question is, why this RK function as the EHM targeting signal? So we did some brainstorming. Maybe this is they are the binding sites for some negatively charged phospholipids, or maybe these are the binding sites for all the membrane proteins. So anyway, we took a reverse genetics approach as well as forward genetics approach, trying to answer this question. So I'm going to be very brief. Initially, we collaborated with Shunyuan from the University of Missouri. We got a panel of mutants that contain mutation in the these genes encoding the enzymes in the metabolic pathways of phospholipids, including PLC, PLD, PLA, and we tested them with powdery mildew. So a former graduate student, Chung Zhang, did this work. So Chung now is a Minus fellow at the University of UC Berkeley. Okay, so Chung found out that PLD data, when it is knocked out, the mutant is more susceptible to powdery mildew, whereas PLD alpha shows, the mutant shows enhanced resistance. So it's kind of interesting, right? And so she later did some quantification work and some other work trying to see whether PLD data is required for RPW8, but the answer is no. So RPW8 function independently of PLD data or alpha. So that we hit a wall. So we then look at the phospholinositides, which are the derivatives of this the phosphatidyl inositol, or PA, right? So the fatty acids chain will be linked through the glycerol backbone to a inositol ring, and the inositol ring can be phosphorylated at 3 different positions. So you have 7 sort of inositides, and these lipids can function as structural material as well as bioactive signaling molecules. And in animal system, they serve as spatial and temporal landmarkers for subcellular membrane and domains. So we thought, okay, maybe the host-pathogen interface, the EHM, might contain some special phospholinositides that may give RPW a signal to go there. So we then tested whether RPW8, in this case 0.2, binds to any particular phosphorinositide. And indeed, a former graduate student, Bob Bucky, showed that RPW8 preferentially binds PI3P. A little bit of binding with PI4P, but mainly it's PI3P. And through collaboration with Xiao at Virginia University, and we showed that RPW8 protein does bind to the PI3P containing liposome, as shown by the surface plasma resonance analysis, as well as the binding sort of association, deassociation contents analysis. We found RPW2 binds PI3P much tighter than to PI4P. But now the question is, what is the role of this binding? We still don't know. So we try to obtain genetic evidence. So former graduate student Chong Zhang then made a dominant negative PI3 kinase, that PI3K gene. And PI3K is a single gene, knocking out is lethal. And so luckily, Chong found a dominant negative form and expressed it in Arabidopsis and found out, again, it is still developed this kind of lethal phenotype, and the meristem will become distorted and later on during the development. So we then later drove this dominant negative form of a PI3K with an inducible promoter, and it developed normally. But when we add the inducer, we found something interesting. Now, this is buffer control. You see RPW off P is targeted to the interface. You see the host-pathogen normally. But when we add the estradiol, which is the inducer to induce expression of the dominant negative form of a PI3K, we found RPW is mistargeted to the plasma membrane, to the maybe vacuole, and its targeting to the interface is greatly reduced. And as shown by introducing RPW5P also in the dominant negative form of PI3K, we found, okay, you have a few EHM localization of RPW5P, a little bit, but mostly it's as puncture, got stuck, okay? But in this case, we thought, okay, in this, maybe PI3P is enriched at the EHM that provides the trafficking cue. So we then speculated that PI3P, using biosensor of PI3P, you should see enrichment of the PI3P biosensor. But initially we thought, yes, indeed, we use this 2x5, which is the domain from an animal system known to be binding to PI3P specifically. But we found, okay, it is there, but not that sure. Until we then used an endogenous PI3P binding protein from Arabidopsis, shown here. So ATPH1 fused with YP, we put into Arabidopsis, we found that it is not in the interface. So suggesting that PI3P may not be enriched at the EHM. So we said, how this RPW with PI3P binding can help RPW to be localized to the EHM. Anyway, we keep that question in mind for a little while, then we talk about the trafficking machinery. I think my time is run. The nano machinery in the universe, and they know precisely where to go. And due to the presence of the regulatory proteins like snare proteins, RAP proteins, and we know snare are complexes that contains QA, QB, QCs, and a V snare, and they can mediate the vesicle trafficking and a fusion between the donor domain and the targeted membrane domain, right? So we thought, okay, RPW might require some of the specific, snare complex for its precise targeting. We used the reverse genetics approach. We did not really find something, but we then performed the forward genetic, study from postdoc Xiao Ma identified 2 mutants, well, many mutants, but when we got 2 characterized. First one called DRF1, defective in RPW function. So DRF1 is a gene that contributes to the H22 production in the interface. So contributing to RPW function, defense function. I don't have time really to go through this. I'm going to skip this. And Shenfeng also identified a second mutant called DRF2. And this mutant shows a phenotype that is like so the branches show burning down phenotype, which reminds us of previously characterized mutant called VTI11. When it is knocked out, it shows exact phenotype. So we surely enough, we found this is a mutation in VTI11, which is a QB snare. Okay, and when it is knocked out, it compromised RPW resistant function. And that QB snare VTI11 is known to function to guide vesicles from the trans-Golgi network to the pre-vacuum compartment, then to vacuum. Okay, so that pathway. We say, oh, how come then RPW8, goes to the host-pathogen interface? Has nothing to do with the vacuum, right? That trafficking pathway. But anyway, we did confocal imaging to say whether RPW trafficking to the EHM is compromised. And yes, indeed, we found that this is like the wild type RPW goes to the interface. You have the rings, means the EHM, labeled by RPW8 RFP. But in the mutant background, RPW8, rarely goes to the EHM, but instead it goes to the vacuum. It actually, partially accumulates in the vacuum. And when we introduce RPW1FP into the VTI11 mutant background, we found, yes, RPW8 is in EHM, but only very, very few hostoria. Most of the hostoria do not contain the EHM with RPW8 labeling. And sure enough, RPW YP shows puncture as well. So suggesting that VTI11 is required for RPW targeting to the EHM. And women's group, as well as our group, found out that if we block the TTN to vacuum trafficking pathway by using the pharmacological inhibitor nitroquinone B treatment, we found that RPW8, even from the naive plants, accumulates massively as puncture. Okay, so this is like very interesting because normally we thought RPW is induced without infection, without RPW8. But now, if you just treat with nitroquinone B, RPW8 shows up immediately, suggesting RPW is constitutively expressed, although at a low level, but is constitutively or constantly removed in the vacuum. If we block the vacuum trafficking pathway, RPW is going to accumulate, then you activate defense. Okay, so RPW is negatively controlled by vacuum dependent degradation. So then we understand now that RPW8, in a wild type background, you see RPW goes to the EHM nicely. So the vacuum membrane is now labeled by a marker, the tonoplast or the vacuum membrane nicely. So Shenfeng found out that in a different mutant background called the ITT3, which also contains a mutation in VTI11, right? So RPW is not or rarely found in the interface, but instead RPW8 RFP accumulates in the small vacuum beautifully there. Okay, but intriguingly, GFP tagged VTI11 is not in the EHM. It is in the tonoplast. As shown here, actually, if you look at closely enough, the EHM is the red thing here, but green is VTI11. So VTI11 is not in the EHM. So this is another perplexing question. Why then VTI11 is required? So at this stage, we tried to look at all the partners of VT11, found out the SYP22, which is syntaxin, the QS there, may be partially localized to the EHM. Maybe a small pool of that is localized to the EHM. Okay, and we did the plasmolysis to show indeed, SYP22 may be retained in the EHM. So anyway, we localized SYP22. Again, the mutant is more susceptible to powdery mildew, and RPW's function is compromised. So taken together, now it showed that looks like RPW is made, but without infection is removed in the vacuum to avoid the costly activation with fins. But in the presence of the host-pathogen biogenesis, RPW8 is switched. There's a transient reorientation of the traffic towards the host-pathogen interface, and RPW goes there. Okay, so VD11 that snare plays an essential role. So this is a model we now use to explain the RPW sorting. Looks like RPW's vesicles is sorted at the TGN, normally to vacuum for degradation. But when the pathogen invades, there's a signal which we have not identified yet. RPW is sorted to the host pathogen interface. Okay, now we then use the alpha-4-2 to see what kind of protein structure RPW has. And if I figure out this, RPW actually forms 4 helices. And sure enough, the RPW domain and that domain also shows exactly the same structure. And we thought, maybe RPW8 somehow now function as a sorting adapter to take the resistosome to the EHM for activation of anti-host-pathogen resistance. So that's the hypothesis. Okay, so without RPW8, powdery mildew is happy, and this reorientation of the traffic to build the EHM. Okay, but in the presence of RPW8, RPW8 can maybe interact with RNRs or TNRs or whatever the resistosomes, right? And takes the resistosome, just provide a traffic signal to take the resistosome to the interface. So whereby you have more, effective activation with fins. So the time is up. I don't know. I have time to talk about MLO. So Professor Chen, if you think now we don't have time, we can stop here to take questions. But if you give me about 10 minutes, I can quickly go through the MLO story. Hello? So hello? Anybody there? Hello? Oh my gosh, maybe there's a disconnection. Maybe wait a second. Hello, anybody there? Professor Chen?
Can you hear me?
Oh, okay. I just don't know if I should just stop here to take question, or I continue with the MLO story for about 10 minutes.
Okay, go ahead.
Oh, okay, now our lab connected 6 powdery mildew species, including powdery mildew that can infect cucumber, south thistle, tomato, tobacco, strawberry, and hemp, the cannabis, which is dama. And in a postdoc in the lab, sequence all the genomes, did the comparative analysis, figured out the powdery mildew conserved genes or powdery mildew unique genes, and we try to understand the how powdery mildew can invade plants. And so we also use the powdery mildew species to study the molecular basis of non-host resistance. Why rice is immune to powdery mildew and rust? It's a big question. We try to, understand that. So when we infect Arabidopsis plants with different powdery mildew species, and you can line them up. Some cannot even germinate. Some can germinate, penetrate a little bit. Some, cannot finish the life cycle, but some can finish the life cycle. So we can use this as a biological sort of systems to address this question. But anyway, we did the genetic screen. We built a triple mutant EDS1 pathosystem, and then mutagenize it. And then we look for mutants that are either more resistant to adaptive powdery mildew or more susceptible to non-adaptive powdery mildew. So we have these two types of mutants, CP mutants or SNAP mutants. But today I'm going to just tell you a little bit about the CP mutants. Okay. So Chong did a genetic screen from this triple mutant, which is super susceptible to powdery mildew. Okay. She found 5 mutants, we call CP mutants, that are super resistant to powdery mildew, which is very, strange to us. Anyway, I'm going to talk about all the details. We found out that all the 5 mutations land on MLO2. So this was how we got to MLO, right? And we know MLO from body work, right? When you knock out the body MLO, now you get broad spectrum resistance against all powdery mildew. And that is pretty much true in all other plant species. If you knock out in whether it's apple or grapevine or cucumber, you get a resistance. So meaning that, powdery mildew as an organism strictly requires MLO from host cell to function to cause infection. But the molecular basis of that is not understood. And also this MLO mutants shows constitutive expression of defense. So suggesting that MLO may be a negative regulator of defense. Okay, so we then has interest in figuring this out. We know the MLO2, 6, 12 in a small clay, they are functionally redundant, and we knock them out by CRISPR. We made a sectable mutant. So this sectable mutant is still immune, not completely immune to powdery mildew, to any powdery mildew. And now what we can conclude is that MLO proteins, MLO2, 6, 12, function as susceptibility factor of powdery mildew. They have nothing to do with really defense. They may be negative regulators, but their requirement for powdery mildew infection has nothing to do with function as a negative regulator of defense. So we look at the localization of MLO2. As you can see, MLO2 is focally accumulates around the penetration site, precisely in the haustorial neck region. Okay, and we found the sectable mutant. When we infect the plants with powdery mildew, the powdery mildew spore can germinate, can even penetrate cell wall 3 times, then stop. They cannot differentiate the haustorial, suggesting that somehow without MLO, powdery mildew will stop differentiation, cannot differentiate haustorial. Okay, so now we hypothesize that MLO proteins function to build this haustorial neckband, which is a membrane diffusion barrier to stop the plasma membrane of the host with the EHM, and maybe also required for really differentiation of the haustorial. Okay, we are doing genetic screen and protein interaction work to try to figure out this. Anyway, just 2 minutes. I want to show that we use this sectable mutant to introduce MLO from body, and it shows it can compensate the loss of MLO2, 6, 12. and also we express the MLO7, okay, from a different, subclade. Using MLO2 promoter in leaf, we can say, okay, MLO7 can compensate the loss of MLO2/6/12, but not MLO1. Okay, if you just express MLO1 in leaves, no, it is highly expressed, but it cannot compensate. But then this made us to really think that we can have a nice, efficient report system to assess which MLO genes serve as host susceptibility genes for powdery mildew in any plant species. So we made this non-table mutant, EDS1 PAD4 with PIN1, PIN2, PIN3, which is super susceptible to any powdery mildew. We also knock the MLO2/6/12 in this background. Now plants show no sign of defense, but they are immune to powdery mildew, to any powdery mildew. But imagine if you say, oh, we have a candidate gene from a strawberry, which is octoploid. You don't know whether it functions as a host susceptibility factor or not. We can put into this mutant background now to say yes or no. If it can compensate the loss of MLO2/6/12, yes, it is a host susceptibility gene. Then you can use CRISPR to knock that. So we think we have developed such a facile report system for identifying MLO genes as host susceptibility factor. Okay, so maybe 1 minute to say I always, because my research started from citrus. I want to go back to citrus, and I even tried to go back to China, but not yet. I hope that this one day that I come back. Anyway, a number of years ago had a young faculty, Pan Ziyong at the Huanglongbing University, through collaboration with a famous scientist, Tom Bisley, to work on citrus, okay, specifically trying to understand the mycorrhizae. And we know mycorrhizae is a form of symbiosis, which is very important for plant because especially citrus does not have leaf hairs and strictly requires the mycorrhizae fungi to get soil nutrients. And to try to understand how the mycorrhizae fungi forms symbiosis with host plants, we use the to identify genes that are induced during mycorrhization. And then, graduate student An Ziyong actually recently identified a sweet protein, which is a sugar transporter, that is specifically localized to the host pathogen interface called the periarbuscular membrane, which is equivalent to the extra-haustorial membrane. And we have evidence to show that sweet 1B may be recruited by the mycorrhizae fungi for sugar uptake. Okay, and of course we are interested in some other things like citrus greening, which is a devastating disease around the globe. And recently I engaged collaboration with the famous virologist at the University of Maryland, Professor Anne Simon, who identified a citrus virus called CYVV, which is can accumulate at a very high level in citrus, like lemon, but it does not cause severe symptoms. So and Dr. Simon thought, okay, maybe we can engineer this virus as a vehicle to produce and deliver small interfering RNA to do whatever we want. For example, we can maybe silence TCV, which is another virus that causes Tristiza, the quick decline of citrus. Citrus screening, or we can target the citrus psyllid, which is a vector that transmits the bacteria, and we can even target the bacteria. Although we know gene silencing does not normally work for bacteria, but looks like it can. Our lab actually collaborates with Dr. Simon. We have some promising preliminary data that we can use the vector, the virus delivered small RNA to inhibit bacteria, the intracellular bacteria. So that's the summary. RPW8 is a nice protein, support my career. And we know RPW8 requires ADL1 for function. It binds PI3P, possibly for correct sorting at the TGN, which also engages the QB snare VTI11. And then we also found, accidentally MLO are truly host susceptibility factors when we have the report system for identifying any MLO genes from any crop. Okay, if you want to collaborate with that, you're welcome to contact me. And of course, I always want to come back to the root of my science, which is citrus, particularly citrus spinning. So I'm going to stop here. I thank all the current and former lab members whose work now I talked. The name are indicated by a blue area. Of course, lots of collaborators and funding from NSF, USDA, and particularly the China scholarship concept, because I have quite a lot of visiting scholars as well. So I stop here to take questions. Sorry for the overtime, just too much time. Yeah.
大家看看有什么问题啊？这个夏老师开始的故事就勾起了我这个当年的回忆啊，所以这个我先插两句啊，我这个考大学考上南京农业大学，报的是果树专业。
You too.
搞得小的时候很少有吃过水果，所以你去报过果树专业，我父亲说你毕业之后。可以到离我们家大概有10公里的一个国营农场去做这个技术员，然后你就可以带水果回来吃，这个就是当时那么伟大的一个愿望。现在开始做这个学果树，然后读大学，然后有其他事情，所以这个肖老师给大家讲的这个自己的这个早年的这个生活和这个科学进入科学轨道的这么一个历程，就告诉大家这个就是说 nothing is impossible 就是 anything possible， 所以就说只要大家认真的去做一心一意的做，特别是看到夏老师跟他的导师说你把这个文章这个tracked 对吧 retrack， 然后我们重来做。所以这种思想现在从我们的现在的学生在对我们的导师这样说的话，恐怕没有几个敢去有这样的这个挑战的能力和精神和决心。所以从这个里面就说大家可以学到很多的东西这个如何去做学问，如何去尊重科学，然后确认这个这个方方面面，然后再把它证明这个东西都是非常非常重要的。所以这个是非常非常优秀的一个报告，非常听了这个开心这个勾起了我自己的这个过去的回忆。所以现在大家看看有什么问题在小梧桐里面这个向肖老师提问，然后肖老师也可以给大家解答。所以大家如果去做抗辩的话，我觉得应该回过头来好好看一下，中间可能有一点点断的，但是应该整体还是比较完整的。
常老师，这个问题是应该从那个哪个地方来哈？我这个谈一谈。怎么进不去？你看。有点卡 ，All right. Oh my gosh. So I have one question from from Wang Souming. How do you preserve powdery mildew in the lab? Could you introduce it briefly. Okay. Yes, I think for any lab, keeping even one Paramildew isolate is challenging because Paramildew is an obligate pathogen. It strictly requires a host plant, a living host plant to survive. So our lab keeps 7 Paramildew species. It is impossible for a normal lab, right? I think we have the infrastructure here to do it, and also we have the will, the strong will to keep it. So we have a lot of growth chambers because I'm the only plant biologist working in this IPBL whole institute, and so that means I have lots of growth chambers, and more importantly, nobody complains because they're all working on human all the microbes. So I can keep them here separately, put into different growth chambers. And because Paramildew are obligate pathogens, that means that tomato Paramildew won't affect the tobacco. The Arabidopsis Paramildew won't affect tomato. So that way I can sometimes even put them in the same chamber. They won't bother each other. Unless we have this susceptible mutant, EDS1, pathosystem with the PIN1, PIN2, PIN3, that is susceptible to all Paramildews. So you have to be very careful if you use the susceptible mutant to do experiment, because it's very catchy. It can be infected by any dicot Paramildew. Okay. So my experience is that if you want to keep one, it's fairly easy, as you have to designate one lab member to, always keep providing living plants to subculture once every 2 weeks for the Paramildew. Then you can keep it alive. Okay. Any other questions? All right, there are lots of questions here. Okay. So one question from Huanglongbing Sloby. Does RPW8.1 work together with RPW2 to enhance Paramildew resistance? Or if RPW1 is redundant? Good question. Actually, Professor Huanglongbing addressed this question quite elegantly, I think. His recent work is focused on RPW1, and together with his earlier work, he found out that RPW2 is mainly expressed in the epidermal cell, whereas Paramildew invades. RPW2 also is expressed in the mesophyll cell, but not at that high level. But RPW1, its expression is restricted only in the mesophyll cells. So it looks like there is coordination between these two isoforms. RPW1 acts in the mesophyll cells to active defense that leads to the production of hydrogen peroxide, which will be, looks like, transported across even the junction of the two layer of cells, providing maybe even like the defense molecule H22 to the epidermal cell. So they have coordination for sure. And so when you have both RPW1 and 2 expressed together, you always have better resistance. Okay. And recently, women's group also found out if you express RPW1 in rice, you can actually have a resistance, enhanced resistance against a bacterial pathogen of rice, suggesting that RPW8 ectopic expression can somehow activate time-perturbed immunity. To some extent now, I think it is true. It looks like the EDS1 pathosystem signaling pathway is required for RPW function, and it is also required for the basal resistance. Basal resistance is mostly comes from temperature immunity. So that means somehow the signaling derived from the PAMP receptor somehow will be channeled down to the EDS1 pathosystem pathway. That is enhanced. That can be enhanced by RPW8. Yeah. So I think now we know that PDI and EDI is connected. Somehow is connected, but the precise connection between the PDI signaling and the TNLs or EDS1 or ADR1 or RPW8 is still not known. Okay. All right. It's a question from 3，请问夏老师明年还在博士吗？要求是什么？ Yes, actually, I just submitted a big grant proposal to NSF, to the NSF genome research program with a number of collaborators. I'm the PI. I think we have a very good chance to get it funded because I think it was reviewed last year. It was very positive. So hopefully we get it early next year. Then I want to recruit at least 2 graduate students. So I welcome everybody to apply. If you have interest to do a PhD at the University of Maryland, yes. I normally recruit students from the molecular and cell biology graduate program, which requires now actually only TOEFL, okay? Not GRE anymore. But you have to have outstanding GPA. You have to have good recommendation letters and also some good research experience, especially publications, to just convince me that you are very productive and you are serious, you're passionate about science. Okay. All right. So another question from 未来可期. Have you tested RPW8 for resistance to all the disease, all the pathogens, such as downy mildew? Yes. More than 10 years ago, Dr. Huanglongbing actually tested some of the RPW8 transgenic plants against the downy mildew called Hyanoparanospora Arabidopsis. Okay, and we found out that the transgenic plants, when RPW8 genes are expressed, they can have resistance against this downy mildew. But the tricky thing is that in order to get good infection from the downy mildew, we have to keep the plant under a very high humidity condition, which actually suppresses RPW8 expression. So you have to play with the humidity. Okay, maybe the first few days you keep humidity very high so that the spores from downy mildew can germinate, and then you have to reduce the humidity level to an extent that RPW8 expression is permitted. Then you see resistance. Okay. All right. Another question from Wang Sanhong. Do you think what main different mechanisms between Rosaceae family plants and Arabidopsis plants? Okay, what's the main? I have to try to figure out the question. What do you mean?
I think it's Professor Xiao. I think he's trying to ask you the question. Maybe on the powdery mildew resistance.
Okay, yeah.
What could it be difference between Arabidopsis and Rosaceae, the 蔷薇科?
Yeah. Well, I guess the work we've done with the Arabidopsis system does have some, I think, implication as to the resistance or susceptibility of rosaceae plants such as roses or apple, they are resistant to powdery mildew to some extent. I believe that some of the RPW8 domain containing genes, they are not sensor NRs, but they can function to enhance basal resistance, as well as the resistance triggered by some other sensor NRs, right? I think the basal resistance pathway should be highly conserved. EDS1, PAD4 should be there as well. So if you knock out this orthologous genes in rosaceae, plant species, you will get compromised resistance against the powdery mildew as well. And with regard to MLO, that is absolutely conserved. If you can identify an MLO gene that is number one expressed in leaf tissue or stem tissue where you have the powdery mildew infection. Number two, you can demonstrate whether it can compensate the loss of MLO2 612 in Arabidopsis using our report system. Then you know that MLO gene must be, serving as a susceptibility factor, and you can really target such MLO genes using CRISPR. And I guarantee you can get your gene edited plants that are immune to powdery mildew. Okay.
There are a couple of papers in horticulture research which has worked on the MLO in grapes and maybe apple, I cannot remember. So you can go ahead to look at the papers in horticulture research.
Actually, in Xiao, Professor Wen Yinqiang in Xibei Nongling worked on that, and I think Yinqiang identified some candidate genes, and his group actually used CRISPR to knock out those genes, and this gene-edited grapevine plants indeed showed resistance, at least partial resistance to powdery mildew. So now the key problem is that not only you need to identify the functionally relevant MLO genes, but also how you're going to avoid the activation of the strong constitutive activation with defense, because the mutant plants, MLO mutant plants will be showing early senescence or reduce the plant stature, and which of course won't be make it appealing to farmers to so we have to get rid of that. You can of course, if you don't worry about all the pathogens, you can in the same time knock out the EDS1 PAD4 in, for example, grapevine, and so you won't have this constitutive activity defense, but you can get maybe complete immunity against powdery mildew. In some extreme cases, maybe you can do that, because I know that in California, grapevine is a big deal, big industry. I heard a talk from a pathologist from California. He told me that over 95 or 85% of the cost the farmers there use to control grapevine diseases is from controlling powdery mildew. So powdery mildew is the major pathogen that really requires so much spray fungicide, and the cost is so high. So under such a scenario, maybe you can develop a grapevine with mutations in EDS1, PAD4, and at the same time also knock out the functional relevant MLO genes. Then you can get nice grapevine plants that are immune to powdery mildew. Okay.
Okay, Xiao 老师, I think it's, you may have a lot of questions, but I think it's, can you answer those questions on the system? And I think we want to close today's seminar. And so thank you very much, 肖老师。我刚刚在开始的时候可能也忘了介绍我自己了。我是陈国明园艺研究的主编所以非常高兴大家来参加我们今天的分享会除了园艺研究之外，我们还有果树研究、蔬菜研究观赏植物研究和这个饮料植物研究，也欢迎大家投稿和阅读我们的文章今天我们报告就到这儿，以后有什么问题大家还可以向肖老师请教可以写邮件向他询问，然后我们这个报告也会存起来，也会回放，所以大家明天应该会收到这个回放的这个信息，好吧？好，谢谢大家，肖老师 ，Thank you very much.
Thank you, Professor Sen. Thanks for the great opportunity, and thank you, everybody. From China, we used to be the colleague in this field. So thanks very much. Welcome to contact me through emails. Okay, I can try to address all the questions. So thank you. Bye.
Okay, good night, everyone.
Yeah.